than it's going to be, because there's still a lot, like 2/3 have to be done, but a lot of it is material that we covered even in the lab, so temperature, how that affects growth, microbial growth, the pH, oxygen. So there's a lot of it that's going to be pretty fast and not very complicated, and a lot of it a lot of it might make immediate sense to you because you've already worked in the lab with different environmental conditions. So that shouldn't be too hard to get through. And then I have about I posted a part 1 for chapter 8, and so it's only going to cover enzymes and enzymatic reactions and how they work. And so we're going to get right to where it gets really ugly. Which I think that's another thing I'm going to look through. So it's going to get ugly, I think, with just a lot of metabolic reactions and all that. And so we'll do that for chapter test 3. But I also think I'll make a decision on how much you really need to know, because a lot of people I talk to say, well, if you have a glucose as the entering compound or chemical, you don't have to worry so much about everybody understanding each single step. Maybe you have a C3, so carbon 3 body that then enters a cycle, and that's what you need to know. But just the major steps. So maybe it won't be as complicated as it looks right now on those pages. And I don't know, has anybody done a lot of glycolysis, Krebs cycle, all that? Yeah, you guys did. Anybody else? Because it's maybe basic biology doesn't cover that too much, right? Yeah, I don't know if anybody took some basic biology classes, but I mean it's part of for some reason it's part of introductory microbiology. I would, I guess microbiology is it's important in any form of life, but I would probably imagine that being taken in some basic or more basic biology class, and then you can move on. But we'll cover the basics, so I'm not going to make you study all the intermediate phases. But for today, we're just going to do this. Should be fairly easy. Enzyme reactions, cover that, and then we'll stop there, and that's what you should study. And I'll give you a study guide this evening, hopefully. I'm optimistic it's going to work out. So we talked about nutrition and nutritional categories. I'm not going to go much into that. We basically just go through until we hit the last slide from last time. We ended up with diffusion and osmosis, and I'll talk about that. But it's clear that every organism needs nutrition to use in cellular activities. That should be clear. And there's macronutrients and micronutrients. Some of them you use a lot, like carbon, hydrogen, oxygen, etc. Some of them you only need a little bit. And you'll find out in the enzyme chapter where those trace elements are important. So they're important in enzyme functions. And enzyme functions are really, enzymes are working, or enzymes are facilitating almost everything in your cell or in any cell. So they're facilitating all these different pathways and reactions for your cell, for any cell to work. And we talked about organic nutrients and inorganic nutrients. Usually organic identified by just carbon and hydrogen atoms, and then those are a combination of other atoms in inorganic nutrients. And these are the 6 main elements that your cell is composed of. There's other ones, but a high percentage is using these 6. We talked about those. And I made a longer detour into exactly explaining autotroph, meaning the carbon source of those organisms is CO2, and heterotrophs they take carbon from some other from a chemical or from a compound that is not CO2, that's organic, and usually made by autotrophs. If something is essential and has to be provided as a nutrient, it's a growth factor, and that comes out that we will see here and there in the next few slides. And we talked about, I don't want to go into that again because we talked about it quite a bit, and it's pretty easy to read up on phototrophs, chemotrophs, where they get their energy from, and then what we talked about, carbon source, CO2, or some other organic compound, and then you combine the two, and you have these different forms of life that use different carbon sources, either CO2 or others, and that can either use the energy from the sun or they use the energy from a compound, from a molecule, the energy that's stored in it. We'll talk about that a little bit more, I think, parasites and relationships in a little bit. So really where we stopped is the transport, and what's important is those are all mechanisms that are important for your cell. So your cell is trying to get nutrients in, it's trying to block certain compounds or chemicals out, it's trying to get rid of waste. So there's a lot of this, and there's active and passive. And what it really means is that passive does not require energy, and active requires energy. And so a lot of times the cell decides, I'm going to spend some energy to get this in. I need it fast, you know. There's probably a lot of active and passive in our lives where we make a decision of letting things just go and they're going to happen, or you need it fast, you want it fast, and you put some more effort in. And so that's a similar mechanism. And so under passive, you usually find diffusion, which is the movement of particles or of a substance from a high concentration to a low concentration. And if you remember the dye in the water, it spreads out. There's no energy input provided, and it still goes from high to low concentration. And then osmosis is really a diffusion, but it's the diffusion of water. So this could be any kind of compound that goes from a high concentration to a low concentration, or that moves around, and eventually you have the same kind of, or a concentration that's equal in all areas. Whereas and that goes for for a lot of different salts, sugar, many different compounds that are water soluble, but osmosis really means water is moving, and it's usually there's two components. It's water, and it's moving or not moving through a selectively permeable membrane, and that would be what the regular cell membrane is made of. So that's a selectively permeable membrane. Some compounds, small compounds, can move in, bigger compounds can't move in or can't move out. So water is really, the diffusion of water has its own name and its own mechanism because it's so important in life. So if you think of like how much water there is in every organism, how much water there is on Earth, life is impossible without water, and every membrane is going to take in water, and also has to, depending on the environment they live in, water might move in, water might move out, and so there's a lot of, it changes the life of organisms depending on where they are. So water is a huge influence, and osmosis explains what happens, and we'll do that next. And then another one is facilitated diffusion, which means you have a carrier that helps maybe to speed up the process, but it's still it still goes along the same gradient. So it's not like you need a huge amount of energy, like in this case where you have energy required and you have active transport, group translocation, and bulk transport. So, and we get to those two. So just stick with the passive right now, and we'll start with that. So we did the one, sugar, I told you in a coffee mug would be the same thing, or any kind of salt. You would eventually have equal concentrations anywhere in there. And it depends on time-wise, you probably, have had a iced coffee and the sugar doesn't dissolve. It takes forever. It would eventually happen, but it's makes the difference because the temperature, the movement of the molecules is slowed down. So you would have to, what I've done before, you take a miniature amount of water, dissolve your sugar in there, then put it in and make it ice. Just in case you want to know. I wanted to open a coffee shop at some point, but I still haven't. Maybe I will. So this is not in your book, and I put that in the evening. I had to prepare that for some other class, and I think it explains it a little bit easier. Or better, the book has some strange graphics in it in some cases, but all it really is, and that you should remember, is the passage of water from a region of high water concentration through a semi-permeable membrane to a region of low water concentration. And it sounds funny when you say high, or when you hear high water concentration and low water concentration, but if you think of this being a beaker, one of those beakers, and it had a semi-permeable membrane, maybe you have like a filter paper in there or something, and you would have a solution over here, and what they call it is hypertonic. So somehow you have to figure out a way how to remember this, but a hypotonic solution usually means it has less solute in it. So you have particles that are not water. For example, here you have I think it's sucrose, so that would be a sugar, and so the sugar is a little bit bigger and it can't pass through here. So in this case, because the molecules are bigger, the concentration of water is higher here, and that's a hypotonic solution. So there's hypo meaning less, so there's less of the solute in there. There's no sucrose in there. I'm just, you'll have to figure out how to remember. There's different ways of remembering how hypotonic solutions and hypertonic solutions, what they mean. Because, if somebody says to you, well, so you have a hypertonic solution and you throw a cell in, what happens? Then you should know what that means. But then if you see high water concentration, which that's what you worry about in osmosis, hypotonic might lead you the wrong way, because it's hypo meaning low or less than hyper meaning high. So it's hyper and hypo is related to the solute, not the solvent, which is water. Now that I have adequately confused you.
Yeah.
So, all you have to do is really find, osmosis is such a major process, you'll find it anywhere in any book. If that confuses you, just find a way. There's different ways to learn it or to remember it. Just find one that you're comfortable with. And so what happens is the water, there's always exchange. So you don't, because water can readily move across this membrane, don't see it as movement of water in one direction. There's obviously, here look, shown by small arrows, there's an exchange, and in a solution that has the same amount of solute in it, so you have an isotonic solution, it's not hypo or hyper, there's exchange that basically makes the solution or the movement of water even out. So you have some going in, some going out, but in essence nothing changes. But there's movement. And then in a situation like this, the movement of water into this side of the chamber is higher than the one out. And it makes sense. The water wants to do the same thing any other compound would do. It would try to basically create a isotonic solution or be equally concentrated everywhere. And it's just in cases like in a cell, or in this case, not always. It creates problems for a cell, for example, because you would have a lot of movement in here, and that would increase, for example, the pressure in here. So you have to find ways to keep that from destroying the cell. And that's why we really talk about osmosis, because it is something organisms have to deal with all the time. So if the water concentration of both sides is equal on both sides of the semipermeable membrane, it's called isotonic solution.
So is it just the pressure that's making the water go out, like in that situation then? Or, I mean, what's making the water go out?
Out of...
If the concentration is higher, I can understand why the water would want to come in. But I don't, why would the water go out?
Oh, you mean, well, because these molecules are always moving, and they're not, it's not like all of it is just directed one way. You still have to, there's all these random movements, and the general movement, or the overall movement, is to equal out the concentration into the right cell in this case. But there is some that's going that direction. It's just a lot less. So there is a difference in.
But is it because of the pressure that's building up in there, or why?
No, it's always.
Just because of random movement.
Yeah. So it's not related to the pressure, and it's just you have different in an opposite, in a hypertonic solution you would have the opposite effect, but you still have some going the other direction. And that's really why we're studying osmosis, or why it's an important thing to know or remember. The movement of water across cell membranes is really what we're worried about, or what we want to know about, and the balance of water between the cell and the environment is crucial to organisms. And so if you, for example, I don't know if that, I'll come right back. If you, for example, and think of, I thought I had that somewhere there, but if you think of, food preservation, when you salt, you salt meat, or you use sugar, those are like highly concentrated. You basically create this kind of environment around what you want to preserve, and so it picks up a lot of the water, and therefore it's so highly concentrated that, for example, it would basically draw all the water out of, number one, draws a lot of the water out of the say the meat, but it also pulls away all the water and creates such a strong pull that it would make it impossible for microorganisms to survive or reproduce. So that's why we use that. So it also has something to do, or it's also based on osmosis. And then if you look at there's a little bit of a difference, and that's you can't see that so much. This is hypotonic, isotonic, and hypertonic. So this one means you have a lower concentration of solute, or a lot of a higher amount of water. So in an animal cell, that could lead, depending on how in our case, for example, we have the skin. We have various protective layers to make sure we're not exposing a cell to concentrations of water that they would lyse. But eventually, you would, or in general, the mechanism is because the concentration of solute in the cell is high, the water wants to move in, and so it leads to a bursting the cell or lysis. And in isotonic solutions, it tends to be normal. And in the hypertonic solutions, obviously the opposite happens. The cell has more, or the surrounding, like the salted meat and the bacteria sits on there, the surrounding is full of solute, and the water wants to move out of the cell, so the cell shrivels. And so it makes sense. It bursts, shrivels. And the same thing happens in the plant cell, but in plants what's really normal is, or in general what's normal is we deal more with hypotonic and isotonic situations than with hypertonic. So that the environment is higher concentrated than our cells is usually not the case. And I mean, if you think of humans, I always think of like, you know, we're basically, and I think that they call it that sometimes too, we basically created our own internal ocean. We have blood, and the blood is concentrated a certain way, and so that just stays. So our blood is basically what we're dealing with. Spider?
Yeah, it was.
We should have. We could have preserved it. There's a collection over here. Who wants to see a tarantula? I saw it yesterday. No. It's
I'll
dead.
really have a hard time sleeping if I do that.
Huh?
I'll have a hard time sleeping if I do
Really?
that. Yeah. Ugh.
Okay, you already have a hard time sleeping.
I know, that's what I'm saying. I'm just curious. Too worried about it.
You should go to Mount Diablo every October. They start walking around. I mean, a lot of people know that. Yeah.
Have one.
Yeah? You do have one?
I had one.
Oh,
They're
yeah.
kind of boring because after you like decided to take pictures, they don't do it.
Yeah. Yeah, there's not that much going on, right? And they come out every 7 years, I think. I mean, at Mount Diablo, maybe they're...
I think they're out all the time.
Really? I thought
Lake Del Val, they're out there.
for mating or something, they come out every 7 years. That's when you see them all over the place. Well, you know, it's not just one generation. I mean, you know, 1 year the 7-year-olds come out, and then the next year the 7-year-olds come out. You know what I mean? They're not just... huh?
They live a long time, though.
Yeah? How long?
They live like 20, 30 years.
Really? You hear that, Jen? They live like 20, 30 years. It's good news.
Not if I was around, I would kill them immediately.
You're not going into general biology, right? Are you going? No, I can see that they're not. I wouldn't be too comfortable with them on my skin either, but I think my older brother was absolutely scared of spiders, and eventually he bought one too. I think it was the way how he dealt with it. Yeah, to like let it walk around. And so in plant cells, it's a little bit different because there is a wall, and the cell wall can actually, in a hypotonic solution, so water is moving in, which is good. The plant looks healthy, and there's turgid, called turgid. There's pressure because water is trying to move in, and the cell wall is slightly pushed out. Under normal conditions, actually, and that creates the flow of water too. That's part of it. The higher water concentration and the higher solute concentration in the plant. And then under normal conditions, it actually looks more like this, so flaccid. And then under hypertonic solutions, you would have water draining out of the plant, and you would usually find the cell plasmolized or dead, leading to a death of the plant.
Is that what they usually label is referring to as the plasmolized?
Yeah, I would say, you know, the thing is, it's hard to. I was trying to make sense about. I was worried you're going to ask the question because no, I you know, when I look through this, I think, how do you explain that an animal cell will in hypertonic solution shrivel, and meaning, you know, it shrivels, and then, you know, what does it mean? Does it die? How do we, you know, how does it usually work? And like I said, there's many other. That would be an animal cell placed in a hypertonic solution, but that's not really the case in most cases. Like I said, we have, when you killed the spider, that's when I started to talk about the internal ocean. So our blood is regulated, and so our exchange, you know, all the exchanges between our blood and our cells is highly regulated, and you won't find hypertonic. And then up here, for example, you could say, well, what happens if you jump in a salt lake? You know, you're not dying because you have all kinds of protections like the skin, etc. But in a cell or a cell on its own, animal cell, yeah, that would definitely lead to death. So we talked about this. I just had that in there. But if you're a tuna in the ocean, what's the ocean? Would the ocean be isotonic, hypertonic, or hypotonic?
It would be low concentration, high concentration of solutes.
So high concentration of solutes in the ocean. Hyper. Everybody get that?
Yeah.
And so those kind of fish are always, they're basically always worried about losing water to the environment. And so a lot of the energy goes into keeping water in, and they do drink water. They really take water in and get rid of some of the salt through mechanisms in the gills. So they have a lot of the salt or the sodium chloride pumps or not sodium chloride, but pumps that get rid of the salt.
Does ocean, would it be isotonic for the fish?
It isn't. I don't know what be interesting to know what the concentrations are, but and you know there's differences, even differences in a single ocean, or what we call a single ocean, because evaporation changes the salt content. But in general, I think our cells, an animal cell is not as concentrated, or the cytoplasm is less. It's not isotonic to the ocean.
So you said that it's got a high salt content, which is the solute, and then it also has a high water.
Well,
concentration.
you mean the ocean?
Yeah.
True, but if you look at the concentration of or the final concentration, it's higher here. The concentration of solute per water molecule, or however you want to say it, the concentration is higher here than in the animal. So water is trying to drain out into the ocean, basically. So most of the ocean animals have to deal with trying to preserve the water in them, or take a lot of water in. And the opposite is true, because in a river system you have fresh water, and so this is where all the solutes are concentrated. So the water is trying to move in, and so these fish don't take up a lot of water, and they actually get rid of a lot of it. What's interesting is that, and I said that at the end when everybody was already out, probably everybody was already out of the room. I still kept talking. When we transport fish, and sometimes probably when you pick them up at the store, I don't know what they do, but when we picked up fish, they would always tell us to throw in salt. So you buy salt, like big bags.
I heard that.
Yeah, and you throw them in, oh, you heard that when I thought you heard that in general. You heard that from me.
I heard you say that.
Did you turn around? You were down the stairs?
I think I was sitting right here. I think I even wrote it down.
Yeah, it was the end of the lecture. But so there, what it does is it basically tries to equal out the concentrations between the fish and the bag you have it in, or the tank you have it in, and so the fish doesn't have to work as hard, and because they're stressed, so it's like a cascade, but they usually get stressed from all the bouncing around and changes of environment, and their immune system suffers, and you can easily get disease after transport, things like that. So you try to give them some salt and make those processes very easy. You just increase the quality of the environment for transport by salting.
If you go to a carnival and you win a fish in a bag, you should drop the salt in there to make it easy for it.
I didn't know that happens. Is that what happens? Yeah? You win a fish?
You win fish.
Where does that happen?
Like a goldfish.
Goldfish.
There.
Oh, really?
Yeah.
So what do you have to do?
You throw ping pong in the right cup.
Oh, really? You can win a fish?
Yeah.
I never heard that before.
Throw a ball in the cup.
That is a carnival. to go across to the bar, get a salt shaker. It should. I mean, it's can't believe.
Take my kids to the bar on the way home, get salt.
Exactly.
Wait, okay, so can you, like, summarize this? So, the fish.
Oh, the fish is just an example for you to.
I know, but it has a higher concentration within the fish.
Yeah.
Makes hypertonic.
But you always, if you talk hyper or hypo, you usually talk about the environment. That's important. Because then you know if you have, so if you would have the three, let's say isotonic in the middle, you always talk about the environment. So if the environment is hypo, that's what we talked about, meaning less solute, so it's more water. Maybe you can remember it as hypo meaning high water concentration. I don't think that works, but you just find I don't know any better way. Maybe there is something anybody has found a little idea how to memorize that. But you basically have higher water concentration, so it moves in and it expands the cell. So hypo is here. And then in this case,
Hypo is high water within the cell?
no, it's the outside. So higher water concentration outside means the water wants to dilute the cell, so it moves in. And then the opposite happens here where you have hyper.
Water moves in?
Yeah, here water moves in. So what happens to this? All right, with that. So just like here, you have a hypotonic solution, high water concentration, water moves in through a semi-permeable membrane. And what happens is this expands because in this case, the solute, the big molecules can't go out, the water moves in, so it expands. And what happens is the plant looks good. There's a lot of pressure. So the cell wall of the plant, like say this is the plant cell, would all of a sudden move a little bit this way, so you have more pressure in it, and the plant looks like that. And then in the opposite, where it's hyper, I got it down here, you have a higher concentration of salt here, like in the ocean. So the water in the organism wants to go out and dilute the ocean. The
one with the tuna.
That would be the tuna situation.
With
so
hyper?
Yeah, the ocean is hypertonic.
Hyper?
Yeah.
I have that written backwards.
Once you go through it, by yourself, on your own pace, maybe in the book, it's not difficult, or it makes sense.
Hyper is the tuna in the ocean. And the rainbow trout is hypo.
The environment is hypo. So you have to basically decide for yourself. Usually what they do is the environment has a higher concentration of the cellular cytoplasm, I guess, mostly, or lower. And you can imagine if you take a cell and you for example, think of all the proteins in the cell. Think of all the proteins in your body, all the enzymes, all the carbohydrates, all the glucose, everything that's in your body, in your cells. Those are a lot of molecules, and the water concentration is usually lower out around you or around the cells. So there's usually a draw into the cells. So usually the environment is hypotonic to your cells. Makes sense, right? Because you're concentrating all these molecules in your cells. So usually our surrounding is hypotonic, and so it's more a question of how much water can the cell hold. But there is, for animals that live in the ocean, it's different because the ocean environment is not like the land environment. You have a lot of salts diluted or washed out from rock, from basically the crust, and so the solution. And then there's very I don't know what the differences are, but I know, you go to the Dead Sea, it's high, and then you have differences in ocean areas. I think there's places where evaporation towards the where you have towards the equator, where you have a lot of evaporation but not so much precipitation, you can get a lot higher, or not a lot, but the concentration in salt differs between even areas in an ocean. But they should always be, in most cases, higher than the one in your in the body. So the ocean is a different environment. I just thought the fish gives you an idea, or maybe a way of remembering how it usually works. But you can use the plants. You can just, I think in general, I would just remember that the cell has a higher concentration than the environment. So the environment is high photonic, and water moves into the cell, or tries to move into the cell. That osmosis is a long, it's not that hard, but it always takes time to get it into your head.
It's not hard till you say the hyper, because you like opposite
Yeah,
of what it should be.
I know. Anybody studied that before, has an idea how they do that? No? Yeah?
Kind of. Well, the way I learned it was in physiology. If you
Yeah.
take a blood cell, like a blood one, in your body, it's normal when it's in homeostasis, regular and isotonic. But if you were to have, like, salt water, it would shrivel up, and then the cell itself would be hypertonic and hypo, and also my teacher, he told us that hypo was one was water fearing and one was water loving. So you imagine the cell to be shriveling up when it's like in salt water, or if it's too much, it'll swell up and then get swollen.
Yeah.
So you just have to remember which one
Yeah.
loving and hating and loving.
Yeah.
I always remember hyper people are really skinny, so the cells shrink. And then for the cell to shrink, the water has to go out, so there has to be less water outside.
If everybody gives one idea out, then I'm gonna be sure we're not, it's gonna be crazy in that when we ask a question about it. There's gonna be all kinds of explanations. But it's that's how you have to do it. You just have to remember it your own way. And it's such a basic. If you think of water as they call it the solvent of life, or it's really the basis of everything. So you should have you should have a good idea of how those things generally work. And you can always ask more and more difficult questions. There's probably animals out there that have all kinds of mechanisms to live in different environments. I mean, there are bacteria that live in a high salt environment. How do they do that? There's different mechanisms, and you can go into those and confuse yourself a little bit more. But you could also just try to say, okay, I'll just memorize. If I see a lot of water molecules and nothing else in there, that's a hypotonic environment. And maybe the fish are already the first level of confusion. I'll take that slide out next. I'm just trying to help you. Just find your own way. Osmosis. I think that's one of the things we should include in a question.
Okay.
In a
Just remember the
Thank you.
high amount of water.
So in the last mechanism that is still a passive transport mechanism is it's called the facilitated diffusion. So we're still, to go back, we're still so you have a little bit of organization. We're still in the passive before we go to active, and so all it is really there are some molecules that are some chemicals, polar ionic compounds that are harder to, they're a little bit more difficult to get across the membrane, water would readily pass the membrane, and so you have carrier proteins, and those are just proteins that are picking up from the outside of the cell whatever compound that is, and then basically releasing them on the inside of the cell. So those are very specific. So they have specificity for a certain molecule. It's not just opening up for anything, and those can be saturated. So eventually, when all these carrier protein sites or proteins are occupied or full, you won't see any more. So it's a question of concentration. If the concentration of whatever needs to be going into the cell is really high, it won't just increase. It's just at some point they're all occupied, and it takes time. But that's just another way of transporting things along a gradient into the cell. So there's diffusion, osmosis, and facilitated diffusion. And this just includes the protein. For active transport, as I said before, if you're a cell, a lot of times you can't rely on the slow and a lot of times inefficient passive transport. For compounds like monosaccharides, amino acids, there's a lot of building materials that the cell needs all the time, and so you really need to make sure they come in at a speed or at a rate that the cell can keep up with all the requirements that it has to maintain homeostasis, which is really what that's all about, or what it's all about. And so it can either transport those nutrients against the diffusion gradient. So now we're not anymore in the case of where automatically without energy input, the concentration wants, or the solute wants to diffuse into the water or into the cytoplasm, but the opposite. So it's against the diffusion gradient, and it can also go the same direction but faster. So you speed up. You say, okay, there's a high glucose concentration. Once you need that in the cell, it would probably diffuse, or another small molecule, it would diffuse into the cell because to equal out the concentration, but it just takes too much time. So you can accelerate that process with active transport. What it requires is membrane proteins, so they're called permeases and pumps, and that makes sense. Those enzymes facilitate the permeability or increase the permeability for those proteins, for whatever the cell needs to transport, and it costs energy. A lot of energy goes into that. So a lot of your cell's energy, or any cell's energy, goes into those mechanisms. It's just like anything else. When you want a process to be faster or more efficient, you put energy in, but you also need to do that because you want the cell to get those nutrients in, or to get rid of. If you wouldn't have those, although it costs energy, you're gonna gain energy from the process and from the whole process. So in bacteria, for example, you get nutrients in the cell, and a lot of times they may actually be able to transport drugs out of the cell. That's one of the things they can do. So they can develop or be resistant to certain medications because they're able to actually get compounds out, and in the that's obviously studied. So you have a way of keeping whatever medication or whatever compound you apply in the bacteria if you want to kill it. And there's one thing that's called group translocation. It's another simple process. It just means when the nutrient goes through a process like this, which it looks a little bit different. I think it's up here. group translocation, what it does is it actually takes up the substrate or the whatever the molecule it transports, and while it is actually transporting it into the cell, it also changes it. So, for example, you know, a molecule that's used for in the metabolism, for example, that is important for the cell to have, can be changed from in its structure, so it can immediately go into a process to make energy. So it's just a conversion of the molecule while it is transported in. So it's really efficient. It costs less energy for the cell to do that, rather than convert that molecule later on, and it's fast too. So that's what group translocations are. And then the third process, which would be all the way back, bulk transport. So you have, in this case, you have endocytosis, exocytosis, and phagocytosis. It's just transport of big amounts or big macromolecules that wouldn't be able to go across the membrane through little protein channels or through other processes. So you have endocytosis. You bring them into the cell through a vesicle. So you can imagine if you have these pseudopods, for example, on the cell surface, they can enclose this, and you have these vacuoles or vesicles that bring the substrate into the cell. And pinocytosis is kind of the same thing, except you would they enclose, for example, liquid, which is also a possibility to get depending on what the cell needs. So there's just a difference between liquid and substances, and substances like oily ones, you can imagine that they do what we've learned before with the bilipid layer. They can actually fuse with the membrane, so they don't really need that kind of mechanism. And then exocytosis would be the opposite, where you just expel from the cell. It's not there, but that's the opposite. You transport waste material out of the cell. So, just to summarize it, and I did that already, we have passive diffusion, osmosis, facilitated diffusion, doesn't need any energy, and you can look up whatever else is in there, but that's the basics. You know, you don't need energy, and it goes usually. There's a gradient. There's a concentration gradient that drives those processes, and then if you have active transport processes, you need energy because there's not a gradient. You need energy to put in to accelerate processes or to get molecules in that you need. And there's the three ones. One is just the regular carrier, and then group translocations where you change the molecule. So it's metabolically useful right when it comes in, and then bulk transport is for large particles and liquids. So what would happen if you have a cell in a glucose solution, and the glucose is moving into the cell through a carrier protein?
Carrier protein.
So you have a protein, that means it's not just diffusion.
It would be
Would it be active?
It would be active.
Carrier mediated.
Yeah, it's active.
Going.
I think
No, everybody.
I think
No,
it's the active
you're confused.
transport, but through phagocytosis.
So what you should look at is this, and I didn't point that out, the cell is in a concentrated glucose solution.
Okay.
So you have a high concentration of glucose. What's the glucose going to try to do? Or is there a gradient between the outside and the inside? higher concentration outside, so would it be a passive or an active?
Okay.
So it would be passive, and then it's passive, and but there's a protein involved, so you have it's not just diffusion, it's facilitated diffusion.
But didn't you say that it is possible to have an active since they want to keep resolving a bit faster?
Yeah.
I mean, that could still happen in a hyper solution, right?
Yeah. What was that called? When it's well, that is actually. If you go back here,
And I think the carrier protein is a good way on that, right?
yeah, I guess, in this question I would just say, I would actually write it differently anyway, but because it's, I would talk about maybe it says if a cell is in a concentrated glucose solution, I mean, it's emerged or whatever, so it's already a little bit confusing, or not specific enough. But I would say it doesn't say anything about the speed. It shows you, it gives you a little bit of information about, okay, the glucose solution is concentrated, and the glucose is moving in, so there's a concentration gradient. Okay, so it's passive, probably most likely. That's all the information you have, and you have a carrier protein, so it's not diffusion, it's facilitated diffusion, and that's all the information you have. Maybe one of those questions could be, I don't know, I'll create my own scenario, and then you can say, I want the glucose in fast, and then you call it active transport. I'll do that for every multiple choice this time. I don't know the answer to this, but I have an idea what I would ask. Let's see, where are we here? 25, we gotta move on. 25, yeah. 60. I'm just trying to figure out if we should take a short break. Yeah? Can you come back at 40?
Yeah.
Okay, 9:40, and then we'll take one.